Good morning, good afternoon, good evening, wherever you're at. Welcome to this Twist Bioscience webinar from poultry to peanuts, leveraging low coverage whole genome sequencing to uncover important traits in chickens and production plants. My name is Paul Doran, and I'm the Director of Business Development here at Twist. Before we get started, just a few housekeeping items. We'll answer all questions after the presentations. If you do have a question, please submit it to the Q&A. There's a button down on the bottom. And then following the presentation, if you have a minute to take a brief survey, we'd appreciate your feedback. Before we hear from our two speakers today, I'd like to go over a couple things at Twist, what we're doing and where we're going. Okay, so the agenda is brief intro, power of sequencing, overview of the 96-plex, and then our new alpha product, enhanced WGS. And then we're going to hear from Dr. Douglas Rhoads on applications in chicken, and then from Dr. Josh Clevenger, applications in peanuts, and then take the questions. So what's unique about Twist? So it's really our DNA writing platform. Our technology enables us to make an enormous amount of high-quality DNA with both flexibility and scale. And writing DNA underpins all of our products and services, and it's how we're supporting our customers in a wide variety of applications. And so if you look at what most folks are doing, they're usually making one oligo per well. Twist is making millions of oligos per chip, and that's enabling all kinds of applications, including things like producing genes, oligo pools, variant libraries for screening, and then NGS, which we're going to focus on today. And then also we're making vaccines and antibodies for biopharma. And then last, which I think is kind of a fascinating application, is data storage. We have collaborations going with Microsoft, Western Digital, and even places like Netflix, where we've taken a series called Biohackers and stored it on DNA. So some of the offerings that we have, we've got the human core exome, we've got fixed panels, custom panels, and we're going to talk about those a little bit today. Actually, Dr. Clevenger is going to talk about a peanut panel that he's just made for the community. Library prep, which we'll address today, reagents and kits, synthetic variety of viral controls, and then we've actually been doing a lot around targeted methylation. For other targeted sequencing applications, we've got the enhanced WGS, which we'll discuss today, liquid biopsy, rare disease, oncology, population genetics, infectious disease, and ag bioengineering. Okay, so the power of sequencing. So when you think of sequencing, most people really are thinking about resequencing. So you have a reference genome, and you're going to essentially map reads to the reference genome. And if everything works out perfectly, this is what it looks like. You get nice even coverage across the reference, and there's no gaps, no nothing. And so the reality is, when you do sequence, it looks more like this. And that's due to deletions. You also have copy number variations, and then additionally, you're going to have a bunch of unmapped reads. And this is really when you're thinking about things like looking for traits or if you're looking for disease in unique individuals or unique populations, a lot of times the unmapped reads are actually going to play a big role in those traits. And so if they're not there in the reference and you can't map them, and you end up tossing those reads, you end up missing a lot of stuff. And so sequencing really is enabling us to really look at what content's there. It enables us to look at markers, both markers and structural variation. And then, like I mentioned, unmapped reads are actually become useful. So today we're going to talk about, mainly focus about our 96-plex library, which has been used by both Dr. Clevenger and Dr. Rhoads. I'm just going to go over quickly how it works and what's unique about it. So it's fundamentally different than other library construction technologies in that there's no fragmentation and repair or ligation. It's tunable for high and low GC content, which Dr. Rhoads will talk about in terms of how he tuned it to go after some of the microchromosomes in chicken. There's little or no automation needed. If you have an 8-channel pipette, it goes very quickly, and you can do anywhere from 96 to 960 samples at a time. So how does it work? So you have a DNA template. You're going to put that into a 96-well plate. It uses 50 nanograms. And then you add these random primers that also are tailed with barcodes and Illumina adapters. So in green is the random primer, followed by a sample barcode, followed by a partial P5 Illumina adapter. This will hybridize, and then we're just using extension reactions, and we control the length of extension based on the ratio of dideoxybiotin and DNTPs. So this will extend, it'll make this fragment right here. Now you've got a biotinylated fragment that allows you to do capture. So after this first step, because you've barcoded these samples, you can then pool them into a single tube, and then from there, run the rest of the assay. So you capture, you wash, now you've got, you want to add the next Illumina adapter, so you use a random primer followed by a partial P7 adapter. This will hybridize, it extends. We use both for both of these reactions, we use high fidelity strand displacing polymerases. And that enables us to get these more full length fragments. With the final step to get the full P5 and the full P7, we'll do a PCR reaction. This particular primer has a plate barcode on it. And in the next slide, I'll show you what you can do with that. So you just do a few rounds of amplification. Now you've got your library. The kit comes with size selection reagents, and you do a size selection to get your library. Most people are doing for low coverage sequencing or SCIM-seq are doing 2x150s, typically with the S4 flow cells. So I mentioned the plate barcode. Essentially, the plate barcode allows you to then pool plates together if you want to run, say, 96 or say 4 of these and get 384, or if you want to run 10, you can go up to 960. The other thing that we're doing, and we're going to, Dr. Clevenger is going to talk about this a bit, is what we're calling enhanced whole genome sequencing. So once you have your library, instead of just doing low pass, we have the ability to do capture. And so you take the same library and split it, and then use our capture probes, do the target enrichment, and then you can either just look at the targeted sequence, or you can blend them together and you know, basically like figure out a ratio in terms of how much you want of the enhanced markers. And so this will help if you've got must-have markers, if you need to read heterozygosity. And so we're going to talk more about that. Okay, I'm going to turn it over to Dr. Rhoads. So I'll do introduction. So Dr. Rhoads is a university professor of biological sciences, affiliated faculty member in the Center of Excellence for Poultry Science, and the director of interdisciplinary graduate program in cell and molecular biology at the University of Arkansas. He joined the faculty at UA in 1990 to establish molecular genetics for the campus. He's published on genomics in humans, pathogenic yeasts and fungi, tomatoes, black bears, toadies, rattlesnakes, and scorpions. His main focus has been genomics in poultry, including male fertility, pulmonary arterial hypertension or ascites, and a variety of bacterial diseases. He has 71 peer publications, and he's got 12 industry reports and 177 presentations and seminars. Dr. Rhoads, over to you.
Hello, everyone. I'm Douglas Rhoads. I'm the University of Arkansas. I've been here for 33 years. My training was originally in human and hamster genetics, but I've slowly moved my work all into chickens because Arkansas is a center for growing lots of chickens. We produce about 1 billion broilers every year within 50 miles of Fayetteville. And so I've been working with the poultry industry on various production trait issues, and we have settled on whole genome resequencing for mapping complex traits that are affecting chickens. I'll walk you through some of how we got to this point. I got interested in helping map traits in poultry, but we experienced lots of different problems with using SNP chips. We tried them. We probably spent $300,000 trying to use SNP chips for working on some of the traits we are interested in. But we then stumbled upon whole genome resequencing, and I'll cover a little bit about that first baby step in a minute. And that led to our need for improvements through using individually barcoded DNAs, because we originally used bulk segregant analysis. And I'll cover a little bit about our current pipeline and some of the results that we've had from that. So basically, we're mapping chicken quantitative trait loci. The trait that I originally got interested in was ascites, which is a form of pulmonary hypertension. Chickens that put on lots of meat, their cardiovascular system is sometimes limited in how much oxygenated blood it can deliver to that huge frame. And if they get behind, then they will crash and develop ascites, which is an advanced form of pulmonary hypertension. My colleague, Dr. Nick Anthony, had developed 3 divergently selected lines. That's what's shown here in the graph. The resistant lines, only 7% succumb to a hypobaric chamber challenge, whereas in the susceptible line, most of the birds die from ascites over the course of 42-day challenge. And those were derived from a advanced breeding line called the REL, and that's the green line, that's the unselected line, the original parent line. And we got involved in this because the SNP chips were coming out as a result of the Chicken Genome Project. Chicken Genome was one of the first non-mammalian genomes that for a vertebrate that NIH paid for. We tried the 30,000 K SNP chips, or 30K SNP chips, the 60K SNP chips. We went through multiple generations, and although we got signals, anything we mapped it ended up being a very minor allele when we went through all the trouble of doing marker-assisted selection. So we got interested, because we couldn't map anything with the SNP chips, we thought we would look for copy number variation. And so we then went to a bulk segregant whole genome resequencing. We had 2 phenotypes, resistant and susceptible. We had both males and females. We generated 2 pools of 10 DNAs each, and we submitted those on a HiSeq-X for 2x125, 666 gigabases of sequence per pool. And the remarkable thing from that was we were able in the REL line to identify 12 million SNPs. So we'd been trying to use 60K SNP chips, and all of a sudden we were at 12 million SNPs. And this just shows some of the data. This shows a region on LRR-TM4 where the males have a predominant SNP relative to the reference, and the females show no sign of that. So this would be a male biased region. And you can see that the region spans more than 50,000 bases over the 3' end of the LRR-TM4 gene. So this got us very excited. We did the whole genome. This was on the earlier version of the genome, the GGA6. And I know Paul wants to talk a little bit more about the quality of the genomes that we use for these things, but we were able to map then 28 different regions. We validated 2 regions. One was that LRR-TM4 that I showed up here, The other was a region on the 3' end of the CPQ gene. I don't need to get into all the details of that, but we then did marker-assisted selection for those two genes for 2 generations to generate a homozygous line for the non-reference SNPs for both CPQ and LRRTM4, took those into the hyperbaric chamber challenge, and that's what's shown in this graph here. The yellow line is the marker-assisted selected females versus the orange line, this is the rel line. The rel line is the yellow, and the orange line is the marker-assisted selection. And you can see that there is a drop from about 24% ascites incidence to 14%. So that's not just a 10% reduction, that's closer to a 24% reduction. The effect in the males was a little bit less, and we were able to show then in a publication that depending on the altitude, simulated altitude, the different genders had a different response based on our selection. So this validated our whole genome resequencing approach, and we then have moved on. So one of the things that was critical for us is because we work in chickens, we don't have a lot of money to spend on per chicken. So we have developed a very inexpensive way of getting crude DNA from blood. It's published back in 2007, where we do a single wing vein lancet stick, collect 10 microliters of blood. We're fortunate that chicken blood, red blood cells are nucleated. We can then triturate that in a hypotonic non-ionic solution, which will lyse the cells but retain the nuclei, which can be pelleted at low speed. We then we'll disrupt the nuclei with EDTA, digest proteins with pronase E for about 2 hours at 37 degrees, and then we can inactivate the pronase at 65 degrees for 15 minutes. And those DNAs are great at -20 for years and years. And these DNAs are directly applicable for any kind of qPCR or other, even restriction digestion. But for making the barcoded libraries, we have found that we can make a 96-well spin plate stack using Paul membranes. We do a 1.2 micron membrane pre-filter to get out any garbage or clots. And then the DNA is captured on a 100,000 molecular weight cutoff filter plate. We can do low speed spins. We rinse then with a low EDTA buffer. We resuspend the DNAs off of that molecular weight cutoff plate in low EDTA tris solution, and then quantify, and those can be directly used then for 96-well barcoding with the Twist Bioscience 96-plex. We have made 6 different barcoded libraries, pooled libraries of 48 or 96 samples each. For the chicken DNA, we tried 1-to-1 AT-GC primer mixtures, but we found that the 3-to-1 gave us much better coverage over all of the chromosomes, and I'll show you where we get that from in a couple slides. The great thing for us is that we had to do the barcoded, or we had to do the pooled samples before, because the cost of making individual libraries can run anywhere from $100 to $150 per library. But with the twist plates, the twist libraries, you're down at $8 to $10 per DNA. And so that allows us to do individual. But for us, we can make them now in our lab. I've had individual graduate students that have made 96 barcoded libraries in 3 hours. And these are rookie graduate students with very little experience. So this is a no-brainer type operation. It works for anybody. We've sequenced originally on HiSeq-X. Now all of our sequencing is on the NovaSeq. And what we've discovered, or what we found, is in our closed populations of chickens, we'll get about 12 million SNPs. But in a lot of chicken work, we're dealing with crosses because the modern broiler is a result of a 4-way cross, and we will get 20 million SNPs segregating. And this just shows that no matter what amount of DNA we put in, we get a good library. And the library, the individual reads span from about 3 gigabases up to almost 25 gigabases. So we don't spend a lot of time making sure that we put in exactly the same amount of DNA from each. We can just clean up the DNA and throw it straight into the kit. Works great. What we have found, though, is that the size profile of the input DNA does affect, and that's what's shown here. I won't bother you with much of the details, but our purified DNAs range from above 10 kb down to about 500 base pairs, and depending on the profile of the size range, we would get different yields from the amount of from the Illumina. For us, probably the biggest bottleneck is the bioinformatics side, but we've got a good pipeline now using FG Bio to demultiplex, Bowtie 2 to align to the genome, SAM tools. We then go on to NGSEP to generate a merged VCF file, which allows us to sort for binary SNPs with sufficient read depth to remove PCR duplicates, because those can be an issue, and with a minor allele frequency that's less than 0.05. We can then process that through SNP test software to calculate the minor allele the difference between the minor allele frequency of the case versus the control, and we then view that in IGV, the integrated genome viewer out of the Broad. For us, using high performance computing, we can do 48 samples that are sequenced at 8x in about 5 days. And we've got a current project where we're going to do 96 samples, and we're going to up the read depth to 20x. And so I'm going to be spending a lot of time in front of a computer for that one. This just shows you some of the data we get with plotting minor allele frequencies for female and male in integrated genome viewer. And if you zoom in on, you find regions like this where there's a male biased region not seen in the female. Zoom in and you can see where the difference in the minor allele frequency between the control and the case is skewed to one side, and that maps. And so we see these haplotype blocks that are segregating where any SNP in here would be diagnostic for that region. So that's the sort of data we're getting out of this. For ascites, we then took and applied this to the whole genome resequencing in 2 commercial crosses. We had 28 regions in the REL. In one cross, we identified 40 regions. In another cross, we identified 25 regions. And not surprisingly, only 1 region was shared in all 3, showing that the genetic background makes a big difference as to what exactly counts as a QTL and what doesn't. And then we spend a fair amount of time doing validation to eliminate false positives, because we've still got some work to do to reduce the false discovery rate. We have applied this whole genome resequencing to identify regions for resistance to bacterial chondronecrosis with osteomyelitis, which is a form of lameness, which is a major animal welfare issue in the chicken industry. And we have a manuscript we're working on the revision from the reviewers just as of today for resistance to infectious bronchitis virus in leghorns. I said that we had to find the right balance. This just shows that SNP density varies between chromosomes. The gray bars are when we looked at the SNP density in REL in our original work, and then we've had SNP density for 3 broiler products And what you see is that you can ignore chromosome 16 for now, because that one is the one that has the MHC locus on it. And so there's only about 6 megabases there. We're missing most of that. But if you look down here in chickens, they have quite a few micro chromosomes. They have the macro chromosomes up here, middle range chromosomes, and then these micro chromosomes. And those have been missing for a long time from this genome assembly. And what we see is that the SNP density, no matter which line we talk about, is much higher on these microchromosomes, and they have a very high GC content. So if we plot by GC content, we get a skew like this for SNPs per 100 base pairs based on GC content. So the genome assemblies are improving, but those microchromosomes represent a different target for when it comes to sequencing. And so our ratio of 3:1 AT to GC on the twist or the 96 plex first strand primers is important. So the conclusions for my part from the whole genome resequencing is, one of the things you do need is a good reference genome. When we were working with GGA5, we were missing so much, then GGA6 came along, and now we're on GGA7, and we've got a lot more of those microchromosomes in there. The beauty of using these kits is that they are species independent. You don't need a SNP chip designed by somebody else. You don't have to make prior assumptions. The reason we couldn't map anything in using the 60K SNP chips was because the population we were working with was homozygous for the SNPs that were on the chip when we did the GWAS. So we could not find the important regions because our population wasn't segregating the SNPs that were on the SNP chip panel. With the individually barcoded libraries, we're getting very high resolution. We're mapping these things at 5 to 10 KB intervals. We can get 20 million SNPs. That makes it very high density. And we're working with only those that segregate in our populations. We don't make any prior assumptions. And our current costs, including the sequencing, are less than $100 per DNA. So it's working great for us. I have to acknowledge that we had funding for a lot of this from USDA NIFA. Cobb Van Tress has provided him funding support, and we have support from the Arkansas Biosciences Institute, which is a part of the tobacco settlement. Everything you've heard from me is my opinion, and it does not represent any endorsement by the University of Arkansas. And although I think you will have great success, the results are specific to my laboratory and not a guarantee that they will be seen in other laboratories or institutions. And thank you for your attention, and I look forward to your questions in chat.
Thanks, Dr. Rhoads. That's really it fits well with the theme the theme of this particular seminar and what we've been hearing over the last 2 to 3 years in terms of people using low coverage sequencing. It's fun to see how the technology has evolved over the years. And you mentioned that you started back in 1990, and I think back then people were using like RFLPs and microsatellites, and now we've moved from SNPs, and now we're moving over to sequencing predominantly. It's exciting times.
Just a little aside, yes, we did try and use microsatellites for years and years, and 200 microsatellites just aren't the same as 20 million SNPs.
Let me introduce Dr. Clevenger. So Josh was trained in the lab of Peggy Osias Akins in plant breeding and genomics at the University of Georgia's Institute of Plant Breeding, Genetics, and Genomics. During his PhD, he was a major contributor in the Peanut Genome Initiative, an industry-funded project to sequence the genome of cultivated peanut, develop genetic resources for breeding programs, and provide improved cultivars to maximize profits and provide sustainability at all levels of the peat industry. After a short USDA NIFA postdoc, Dr. Clevenger worked for Mars Wrigley on the global nut science team, where he designed and deployed genetic improvement projects across 5 continents where peanuts are grown. Clevenger joined Hudsonalpha in the spring of 2020 as a faculty investigator in the Center of Plant Science, Sustainable Agriculture. Specifically, Clevenger's lab is focused on optimizing computational pipelines to optimize the use of next generation sequencing and genomics in genetic improvement projects involving crops with complex genomes. The lab then seeks out improvement targets that are crucial for farm profitability, develop tools for improvement of those targets, and then uses advanced breeding techniques to rapidly deploy new varieties that maintain yield under the threat of pest pressure, stress from extreme temperatures and drought, and mitigate aflatoxin contamination. Josh, over to you.
Thanks, Paul. So today, I actually have the privilege. I was a little bit frazzled because I was in the classroom all morning. I've been in the classroom all week, and I'll speak a little bit about what we're doing down here in Southern Alabama. But today's presentation is about this incredible story about what low coverage sequencing is doing for peanut, a relatively minor crop. And it's a story of collaboration and trust and sort of pushing ahead and trying to do new things. And it's been pretty special the past year, what we've been able to do. And so it's all about peanut, what we've been doing the past year. And really all that work is facilitated by what we call KUFU, which is a project in my lab. And that's the team there that I'm showing that it really makes the engines run. And sort of how we've utilized that across peanut and in collaboration with Twist to do some amazing things, which actually I'll touch on later. Just last night, I got a shipping order of our first panel that we're going to start using. So because it's all about peanut, I will speak a little bit about peanut, all of the a couple fun facts. One of the things is that we call it a segmental allotetraploid, meaning that the subgenomes of peanut, the A and the B subgenomes of peanut, are relatively diverged recently, about 2.5 million years ago. And so the gene-rich regions on the ends and some of the chromosomes actually will interact quite a bit and form these sort of hybrid tetrad structures, which leads to tetrasomic recombination in those regions, even though most of the genome behaves like a disomic polyploid, meaning it's segregating more like a diploid. It's a relatively large genome. It's not huge, but it's also not small. It's about 2.6 gigabases, and we have about 20 chromosomes. Peanut is the daughter, I guess, of Arachis epenensis and Arachis durinensis, 2 wild species of Arachis that diverged about 2.5 million years ago. The polyploid event happened so recently that actually it's really hard to date, and it happened some point less than 10,000 years ago. What that did was it created this extreme genetic bottleneck. It probably was a single population where this happened and then was spread around. And so there's very little diversity between tetraploid peanut accessions. And that causes a lot of issues when you're trying to use sequencing to genotype and do other things with peanut, because a lot of those similar regions pile up and confuse you and give off a lot of false calls. So there's been a lot of struggle with that, which is what I spent a lot of my PhD and postdoc and early career trying to solve. So this is a really fun story. So I wanted to tell this story. This was told in the Bertioli et al. In 2016, which I played a small part. And this was the genomes of the wild ancestors, epenensis and durinensis. And basically it talks about how peanut came to be. And it's really sort of interesting because peanut essentially plants itself. It's geocarpic, meaning that the seeds develop underneath the ground, even though the flowers are pollinated above ground. And so there's not wind dispersed, it actually just plants itself. That's actually why it's highly speciated, because populations tend to become isolated. But also what that means is that it really can't climb hills, right? It can only go down through water, but it also can climb hills if someone is physically taking it of those hills. And also a really funny story from botanist Charles Simpson told me, pigs actually eating the peanuts and being flown around countries and then going their pig pen. He collected many Arachis species on the side of pig pen. So that's sort of the way that they can travel. The closest bee relative to Ipanensis, Arachis magna, is 500 kilometers away from where Arachis ipanensis was collected at lower altitude, meaning it's pretty unlikely that it somehow traveled uphill. Arachis ipanensis and durinensis overlap with the sort of Arachis hypogea, which is the tetraploid version of peanut in that center of diversity, which is uphill of that. And so the hypothesis really of how that happened is that the bee genome species had been actually brought with the folks that were living there up, and then it hybridized in some sort of common garden situation. And when they had unreduced gametes, and then when they collected the seeds, they're like, oh, these are much bigger, and so spread that sort of tetraploid. So really pretty interesting hypothesis and potential story of how peanut arose. So switching gears into like really what I'd like to talk about which is, low pass whole genome sequencing as a breeding student, as someone that was interested in breeding for an international company that wanted to, quickly solve problems across, as Paul said, 5 different continents where peanut is grown. Genomics for me is, when I was actually doing my master's, my early PhD, genomics was kind of like an end in itself. A lot of students were doing projects where they would just sequence a lot of germplasm and do some analyses and things like that. But now, really, it's just another tool in our tool chest to unlock innovation. And so when you think about it that way, you can start thinking about how sort of highly multiplexed library prep can really make a big difference in all of the science that we're trying to do. So if we look at sort of genotyping strategies, and Dr. Rhoads did a really good job of sort of talking about inefficiencies, but really, arrays, which I developed 2 of them for peanut, they're fixed markers. So if your population doesn't work with the markers that are on there, then that's not going to help you. Mapping is really mid-resolution with arrays in most crop species and animal species. You have ascertainment bias, which is sort of the bias that is associated with SNPs that were discovered in small populations that are then trying to ask questions about larger populations that are diverged from those small populations. Basically, it's de novo blind, meaning you get what you get, and anything rare that is not on the array, you're not going to be able to assay. And then at this point, it's the highest cost. You have to have a number of samples that you're going to run on that array to even manufacture it. And then if you don't run a huge amount of samples, it's really not cost effective. But you can do things with it. You can use it for the genomic selection and background selection and things like that. So, GBS and different capture-based assays, or this sort of genome reduction type of approach became really popular. It's going to be cheaper. You're going to have, and in some cases, if it's GBS or some sort of restriction enzyme-based method, you're going to have some sort of ability to find de novo markers, but you're still sort of looking at markers in the same regions all across when you do your projects. And so still has some ascertainment bias potentially, depending on how you designed your probes for capture. And it's, again, pretty low resolution mapping. And so, as a breeder and as someone who's interested in genomics assisted breeding on multiple levels, and wanting to, especially in peanut, where there's actually high value traits that we can bring in with functional markers, right? We don't necessarily have to just go full tilt genomic selection. that's really the thing that we want to move towards. And so that's been my sort of journey over the last couple of years, because you're looking at, depending on your system, peanut again doesn't have a lot of variation, but hundreds of thousands to millions of markers. You're really mapping at high resolution, which I have a good example of. You're absolutely capturing de novo variation in your population of interest. At this point, it's a pretty low cost comparatively. And then there's ways to do imputation that are independent from panels. I mean, I think, the idea that you absolutely have to use a reference panel to have high quality imputation is not true. And then this really cool technology, which Twist has just sort of launched, is this enrichment panel compatible WGS, which I just love to death. And we'll talk about what we're using that for. So obviously I'm biased towards low pass WGS. So I'm just going to touch real briefly about what Kufu does. So Kufu really was launched because we had this great informatics that we developed to do our own work in peanut. We started working with collaborators and started realizing that we needed a really good way to work with folks at a large scale and to help them out with their projects. And so really at this point, we, in our collaborative work and our fee-for-service work, we'll take in tissue or DNA. We will prep the libraries. We use exclusively at the moment Twist 96-plex libraries. We conduct a low-pass sequencing here at Hudsonalpha, which I didn't talk about. We have just this incredible sequencing capacity and really talented folks that can get the most out of the sequencing machine. So we have all that in-house where we do that. And then really Kufu is about our sort of approach to mapping and filtering, again, developed over 20 combined years of thinking about how to do this in peanut, which is a pretty complex system. And so then we love doing things like QTL-seq. I adore QTL-seq with single samples. That's why I love low pass. But again, we work with people where you're looking at, they need genome-wide genotypes for other applications, for breeding and selection to run their models, things like that. So we can do that stuff. And we do imputation if needed. Again, it's all reference panel free, but we also can use a reference panel if needed. We also can impute arrays from sequencing data, which we've seen really good results on. So there's just loads of things that you can do. And I should add that if you really like an array and you're running it, we can impute that array a lot cheaper and have call those markers really accurately. So it's kind of like a new way to think about using arrays. And then for us, we're just really interested in using it for rapidly improving cultivars. So let's go through like sort of the journey of peanut real quickly. It all comes back to, as someone that was interested in breeding and working really closely with the industry of peanut, and then of course working in a global company that's really interested in peanut, which is in their two largest brands, Snickers and Peanut M&Ms. You get this sense of dread of these emerging threats that seem to strike so quickly, right, and have a profound impact on the ability of folks to feed their families, to farm, and to provide high quality food for all of us. So this is one in peanut I'm not going to name because we're still sort of working through it, but this is a really, really good story and one of the earliest ones that we used low pass on. So essentially, we ran a large population. These were actually single plant F2s. We took phenotypes and mapped this particular resistance to this particular disease. Then we did something that was only possible with this technology. And we said, hey, give me another population that's unrelated and give me the best 48 and the worst 48. And so we sequenced those and validated that this major resistance was where we thought it was. We had all this great information, so we identified the variation in a gene that we then assayed in multiple diversity collections that were phenotyped for this trait, and it essentially was able to select for 0 infection across 4 years of looking at these collections. So these are not structured populations, but actually collections. So we believe that we have the functional variation that confers this resistance, right? And this is all, I should go back, this is all just one F2 experiment that led to this, right? So F2, single plants, we sequenced them, we identified functional variation, just amazing. thing. Never thought that would ever be possible in peanut. So the other thing that I love about it as a breeder that's super cool is any of your populations, really, you have genome-wide markers to start looking at the percentage of your current parent or the parent that has the agronomic traits that you're interested in. And this is that one population that we validated in. You can see this massive sort of variation, and these are just the peanuts that were resistant to this disease. So you could see like very easily that, okay, I'm going to select those ones that are resistant and have 80% of my recurrent parent. And right there, you're ahead of the game because you have this information. It's just kind of like a really great thing, really great tool to use in breeding programs, which is what we're doing now with collaborators. So I'll touch really quickly on this. It's really not a project I lead, but I help work on. This is a project led by Daniel Fonseca and Pegasus Akins in the USAID Peanut Innovation Lab, where we helped genotype a huge collection of diversity across the whole continent of Africa. They selected 300 lines that maximize that diversity, and then we resequenced those using Kufu. And so what I'm building here is a story of what we're getting to the end, which is just in one short year, we generated all this useful data. And so we actually did that with collaborators all over the world this past year. So we had collaborators in Argentina where they have a diversity collection that we sequenced, obviously breeding programs in the United States. And so material that was relevant in their programs. Africa, which I just mentioned, there's a diversity collection in India, and then really great collaboration with with the peanut breeding program in Australia, where actually they have a population where it's segregating for a lot of quality traits that they phenotyped, and we're able to develop sort of haplotypes to select for different quality traits. A really good tool for early generation breeding. And not only that, but we've been in peanut really sort of this great work by a bunch of folks at the University of Georgia in South America and Africa to look at, interspecific lines. So actually like bringing in wild species alleles that have this incredible resistance to different diseases. The picture I'm showing you is an elite cultivar, Georgia 13M, and then a new line that is CB1, which is completely resistant to fungal diseases. And so this is an unsprayed test. That comes from wild species. And what I'm showing on the left is a bunch of lines as a part of about 500 lines that represented 6 different diploid wild species that we sequenced with low pass. And the graphs are just a way to illustrate how we can track those integressions. So we score them as 0 or 1. 1 is wild species and 0 is cultivated. So the different lines with the different integressions from different wild species, really great tool to do that. It's easier to do that with low pass than on arrays, obviously. And so again, we were able to capture all these diagnostic SNPs that we could put in a panel. We have in this panel that we developed and just was delivered yesterday, so excited about it. We have SNPs and genes from every diversity collection of Arachis in the entire world. We have functional variation for dozens of quality traits, resistance and stress tolerance through projects we've been working with folks this past year using this technology. And then diagnostic SNPs that can track integressions for 6 different important wild species and probably more. And so the enhanced WGS, which I had the privilege to pilot with Twist when they were working on it, is a pretty simplified workflow. Paul went over it, but essentially after you make a sequencing library, you can enrich it for the probes that you are interested in. And we did a pilot and we looked at, how well does it work? It works like fantastically. I was pretty shocked actually at how well it worked. You're recovering close to 100% of your targets, even below 1x coverage sequencing. That's across all your targets. So you basically have no missing data in your targets across your samples. And that's sort of in genes where I'm showing depth over the baseline coverage of the sample, and also outside of genes. And then we looked at SNPs that we had called in using regular twist 96-plex that had low missing data. So like 10% of 384 samples had no call, and high missing data where 10% had a call. And so a extreme difference in terms of like the sort of regular WGS. So this technology works really, really wonderfully, and that's why we developed the peanut panel. And I'd like to say real quickly that the classroom that I'm coming from is actually a integrated sort of classroom experience with the folks in the Wiregrass region of Southern Alabama, where we're actually doing genomics assisted breeding with them, where they're going through those processes, and they're going to use this panel to actually make selections of the lines that will be back crossed to go in the next generation. So already sort of this new technology is out in the hands of teenagers, and they're learning about it, understanding sort of how to do that. And none of that would be possible without sort of all of these tools and low coverage WGS, and how, peanut breeding has been enhanced with these tools in just a short 12 months. Just so excited about the future. And, peanut is not corn, it's not wheat, it's not soybean. And so just, one group of highly collaborative people with a great product, we really have come a long way in a short time. So these are my main funders, and I will stop talking now.
Thank you, Josh. That's it's amazing how far things have come, and that kids, teenagers, high school students are doing stuff that we only could really never dream of 5 to 10 years ago. It's really truly amazing. Okay, we're going to open it up to questions. If you have a question, please make sure to ask it in the Q&A. And I'll start it off with a comment and a question. So I was at AGBT Ag just I think it was about a week and a half, 2 weeks ago, and Wes Warren presented the new chicken genome, and it was they've implemented all the latest technology. There was a lot of stuff on Revio in PacBio. There was stuff on Oxford Nanopore, and the ability to go telomere to telomere on reference genomes. And the fascinating thing is that the new reference genomes that are coming out for chicken have gone from 34 chromosomes to 41. And so this is really kind of it's kind of a softball question, but like essentially now you've got better references, and what is that going to do for you guys when it comes to using this type of technology? And Josh, I'll let you go first, and then Doug, you can go next.
Yeah, well, what it means is that we can now genotype. We can identify functional variation faster to help solve problems that are really important faster. So we can close that gap with how long it takes to react to emerging threats and other things that we need to do with our crops. And we can do that with pangenomes, right? So that's what we're sort of putting into place now, and really sort of ramp up the resolution. And so my colleague Justin Vaughn, who I think very highly of, always talks about we're getting to almost like the end, right? Where we can. It's no longer a question of what you don't know. It's more a question of how do I do this experiment better, because I can see everything. I'm all knowing essentially. And that's just like an amazing thought when you're out there in the world and you see the struggles that folks have. Like for me, aflatoxin is a big one, and you hear about them, or viruses. We work on a virus in Africa, groundnut rosette virus. I heard this year that folks lost their entire farms, right? So these are things that we want to solve for them, and we don't want to solve them in 20 years. We want to solve them in 5 years.
Well, it certainly, just like the SNP chips were missing diversity in a lot of regions, we always feared that we when we were trying to map the ascites traits, where all of the selection strategies had said there were major genes involved. Should be very easy to map. Turns out there really aren't any major genes. You can just do selection across multiple chromosomes pretty quickly. And I was always very concerned that we weren't mapping anything because our genes of interest were all on the microchromosomes. And we had no idea, nobody could know why they weren't showing up in any of the assemblies. They just weren't there. And that still is the case, that they don't seem to sequence or assemble nearly as well as the macro chromosomes. So Wes has been beating his head against that wall for a long time. And the question that came up every year at the poultry genome workshops was, why don't we have the microchromosomes? And so Wes has solved that issue for the most part. I think we still have a ways to go before we're going to have T to T for those microchromosomes. But certainly with nanopore improving their read, their quality scores, the ability to generate a reference genome for your organism of interest, high quality, is getting better and better and cheaper and cheaper.
All right, thanks. I think we're at the top of the hour. We have time for one more question, I think. So the next question is, what when you're doing low coverage, basically, how do you deal with heterozygosity? Josh, if you could take that.
Yeah, of course. So this is a question I get a lot. There's a lot of folks that are working in systems where they want to know their heterozygous calls, and the short answer is that we're not using a true PCR-free. This is not a true PCR-free WGS approach that is like completely random. It's pretty darn close, but it's not. And so when you talk about a coverage of sequencing 1x or 2x coverage, you are capturing a lot of your sites that you're calling SNPs on. You're capturing more than one read, and so we actually call heterozygotes quite well, and we were able to test that again by looking at samples that were run on arrays, and then we sequenced and we looked at how this was in heterozygous individuals. We looked at how closely we were, and there's a little bit of error where you have a situation where you don't get that other allele, but most of the time, actually around 97 to 98% of the time, you're calling those hets accurately.
Okay, thanks. And then there's another question that Doug, you said you take any recommendations on getting the most uniform data from 96 samples?
Yeah, we've always relied on Herc's 33258 fluorescence rather than spectrophotometry for quantifying our DNA. And if you really want uniform coverage across 96 samples, you need to be measuring the amount of DNA accurately. I think if you look at the system of the 100,000 molecular weight cutoff capture that we've used, that's going to help with having uniform cleanness, because we spend a lot of time doing trying to do organic extractions and ethanol precipitations, and with graduate students they were losing 20 to 25% of the samples trying to do organic extractions. We don't lose anything now. Every sample comes through, but we haven't bothered with getting the same amount of DNA into every one of the first strand reactions. And if we did that, I'm sure we would have very uniform coverage across all 96 samples.
Okay, thank you. Well, so with that, I think we need to wrap it up. We're past the hour. Quick reminder, if you have a minute to do the brief survey, we'd appreciate it. Thank you, Doug and Josh, very much for the awesome presentations, and have a wonderful rest of your day.